This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1236, recorded on July 18th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York, Daniel Griffin.
Hello, everyone.
Let's see, today is Friday, so we have an STD bow tie, and I'm guessing it's trichomonas.
You got it. I didn't even have to give you any hints. I had a whole bunch of hints lined up.
This
one won't be easy. It'll be tricky,
I see the white background with the purple stain, and I've seen that before, so you're going to have to get more bow ties.
I actually have to do that, yeah. And maybe as the listeners might know, so I have a little bit of like an issue with the bow ties. In particular, I think would be the description. So I don't like actually pre-made bow ties. So I get the ties and then I have them custom made because I like them to be a little bit thicker and the right length and all that,
You should have been a pediatrician.
I did think about that. All right, let's jump into it. We got a lot to cover. I don't know if people are aware, but we're recording a Friday morning instead of our usual Thursday night right before I go sailing. And I think it's because, Vincent, you were having problems going to and from Canada.
Yeah, I went to American Society for Virology meeting this week, which is fabulous. 1,400 virologists. It's just great science. Took me 3 times earlier in the week to get there, and then last night the flight was delayed. I got home at like 11 o'clock, and so I said, Dan, let's do it in the morning. And so here we are, because we don't want you to miss your clinical update.
All right, well, let's jump in. Maybe this is appropriate with how difficult it was to get to and from the meeting, but this is a quotation from Napoleon Bonaparte. The battlefield is a scene of constant chaos. The winner will be the one who controls that chaos, both his own and the enemy's. And I just feel like maybe we're in a little bit of a chaotic time as we continue to see a rise in measles cases. A little bit of a change on the cadence here as far as when we get the updates. So, this page is now updated from the CDC every Wednesday, and that's been the case since the end of June. There's been a few sort of changes there at the CDC, if anyone's paying attention. But as I pointed out, I was recently on an NPR, Vincent. I don't know how much longer NPR has. I think there's been some decision to cut. I like the way they refer to state-sponsored media. Cuts to state-sponsored media. I think it's really like support for the arts. But anyway, that's,
you know what, Daniel? Microbe TV survives without government funding, so they can too.
Okay. So, all right. As I pointed out there, and this, here it is, July 2025. Already more measles cases so far this year than any other year since the virus was declared eliminated in the United States in 2000. Was the virus declared eliminated? Was the disease declared eliminated?
I would say the disease, because you can tell if there's disease, but unless you do extensive surveillance, you don't know if the virus is there or not.
Yeah, I think that's a good point. Like, because, that's been the quotation, the virus declared eliminated. But yeah, it's the disease, right? Because, there's always people coming back into the country. And, there's always people coming back into the country who acquired measles somewhere else. But then we've always had this wall of vaccination. I'll talk a little bit about that. But one of the things I want to point out as we go through these numbers here in the U.S., CDC is aware of probable measles cases. However, they're only reporting confirmed. You got to actually get a test. And when we talk about the map here in a moment, you'll see why that seems a little bit suspicious or low, or the fact that we're missing. Maybe there's certain places in our country where people will not go and get that test. But as of July 15th, that was my birthday. 1,309 confirmed measles cases were reported by 40 jurisdictions. Thank you for wishing me a happy birthday the other day, Vincent.
Yeah, you're welcome. You're only 45, right?
Yes. No, 48, I think it might be, actually. My mother, by the way, still owes me a phone call. My middle daughter was harassing her. Mom, a text is not adequate on your son's birthday. Just get that out there. All right, so we've got this wonderful map, and this is this map of So I mentioned we've got over 1,300 confirmed cases, but then you can look at a map and actually see where are these cases clustered. But then you will notice there's a few states where there's 0 cases, right? So we've got it in 40 of the 51 jurisdictions. We've got a lot going on in Texas, a fair amount in New Mexico, Nebraska. Is that Kansas right there? Is right below Nebraska? That's another hot. But you'll notice Idaho, Nevada, Alabama, Mississippi, West Virginia. There's no reported cases, no confirmed cases. So,
but I bet there's virus circulating.
Well, you see, all around, right? There's reported cases, but so it's sort of when you see cases with 0, right, then it makes you a little suspicious, right? We got
because viruses don't observe state borders, right?
Apparently not. And up there in Canada, where you just got back from another 120 new measles cases, just the last sort of week 27, which brings us only up to July 5th. That is 3,822 measles cases. So quickly approaching 4,000 there. So just, keep reminding our clinicians out there, but also our patients as well. Remember how this presents, the 3 C's and the 4 D's, right? So those 3 C's, cough, coryza, conjunctivitis, then 4 days of fever, then you get the rash. So just keep that in mind. You're coughing, you got the really runny, flared up nose and irritated eyes, and then the rash comes. So you're contagious before that rash even shows up. All right, bird flu, right? That's pretty much gone away, right, Vincent? They've updated the reporting. They've streamlined the reporting. Well, okay. I was spending some time this morning. I remember we used to get these nice weekly what was going on, how many herds, how many poultry flocks. Well, at least now we have from the CDC a little bit of guidance for clinicians, clinical guidance for evaluating patients and treatment and post-exposure prophylaxis of influenza A, H5N1. So here we have just a couple key points. I'll leave in a link. But one is basically clinicians should consider the possibility of this as a diagnosis of the H5N1 in patients with signs and symptoms of an acute respiratory illness or conjunctivitis if they've got that relevant exposure history. So sort of exposure to the dairy or maybe the birds in some cases. Patients with relevant exposure histories who do develop signs and symptoms consistent should be referred for prompt medical evaluation testing, and go ahead and start that Tamiflu, that oseltamivir, while you're waiting for those results. And then also think about other folks, so the household contacts, and consider jumping in with post-exposure prophylaxis with Tamiflu for those folks. It's it's relatively low cost, really minimal side effects. I think some people we refer to as the tummy flu pill because it can throw your tummy off a little bit. But other than that, pretty well tolerated.
Tummy flu. I didn't hear that one.
You never heard that? Here's some tummy flu pills. Why do you call it that? Call me in 2 days. You'll know why. And they're like, yeah, dude, my stomach felt a little bit off. So yeah. But it's it doesn't really mess with the microbiome or anything. It just gives you that little bit of stomach thing. All right. Well, here's a big thing that I didn't actually realize the full extent of this, right? So we've been talking for a while about highly pathogenic in avian influenza, right? But, the cows, milk production down a little, but it's not like the cows get really sick and die or anything. Apparently that's not true. So here we have the article, The Impact of Highly Pathogenic Avian Influenza H5N1 Virus Infection on Dairy Cows, published in Nature Communications. So here they looked at the impact of the H5N1 virus on an affected herd in Ohio, right? So let's look at it at this herd in Ohio and then get kind of a sense of what's the deal. So clinical disease, basically cows being sick, lasted for about 3 weeks. And about 20% of the cows had clinical disease. We're going to see seropositivities a bit higher. So the milk losses were about 900 kilograms, liters per cow recorded in infected cows during a 60-day post-outbreak period. I'm converting that 90 kilograms to liters, so it's a little bit off. But seroprevalence was 89.4%. 76.1% were subclinically infected, right? So that's sort of an important distinction here. So about 90% of the herd ends up infected, but we're only seeing, as I sort of said, about 1 in 5 showing symptoms in the herd.
But they don't say what the symptoms are other than loss of milk. Is there anything else?
Well, as we see, and this is what really threw me, clinically affected cows presented an increased risk of death 6 times.
Okay.
Yeah. And so this is interesting. And they say of premature herd removal 3.6 times when compared to non-clinical cows. And go into what's going on with that. So economic losses, which is really what they're calculating here, due to decreased milk production, mortality, right? I just pointed that out. I was not aware of the significant mortality here. And early herd removal were estimated at $950 per clinically affected cow. So almost $1,000. And this one herd total cost $737,500 for the herd during just this observation period. So they say our results demonstrate a production impact lasting at least 60 days, post-clinical diagnosis, and major financial consequences of the highly pathogenic avian influenza H5N1 to dairy farms.
So I misspoke. I said symptoms, but of course, cows can't tell you anything.
No, they
so there's
can't
signs.
talk.
There's
No
signs of
talking.
infection.
Signs. Actually, so you are right. So, so they'll end up with signs. They appear sick. And then, this was really something I did not really appreciate. Infected cows 6 times more likely to die. So 6.8% of the sick cows, so 53 of the 770 sick cows, so about 7% died or were euthanized, right? So they got so sick that they basically, put them out of their misery within, on average, 13.6 days after the diagnosis. They get infected within 2 weeks. They're gone. Another 31% were culled within 20.6 days after diagnosis. So I think that's, you got to be careful. Don't just look at the first 1 to 2 weeks. But it looked like some of these cows just were doing so poorly. So I think that's a big thing with the cows. They look sick, they can't produce milk, and you're losing quite a bit of your herd. So this is not, as benign, I think, as we sort of thought about maybe before, 10% milk reduction for a period of time. But you're losing you're losing cattle. You're losing cows here.
I wonder if these cows who die have some comorbidities that we don't know about.
Yeah, most of them are probably chubby and they don't keep their hair dry. Yeah, no. I mean, I'm glad you bring that up because that seems to be this modern thing, and I've been pretty amazed at how resisted. I was having a conversation. I don't know if I shared this before. A friend of mine works with some people who are just not willing to believe that disease kills you, and so there was a discussion about, I think it was influenza or COVID and children dying, and they're like, well, were any of them overweight? And it was just this, really, because if you're overweight, that kills you. It's just this sort of absurd idea about blaming people for things. And
so
all right.
They say disease doesn't kill them. So what kills them?
I don't know. This is like the gun and the bullets and, people don't kill you, it's whatever. So, I think as the catch line of this show, viruses, the kind that make you sick. It's not like the show about wet hair, So. All right, you changed the tagline. All right, so talking about flu, the big update I wanted to do here is, as we've talked about before, it takes a little while for all the counts to come in. And the big count that I wanted to point out, because, there were a lot of discussions early on, and I love that article, COVID is bad, comparing it to flu is worse. Flu is bad. And a total of 260 influenza-associated pediatric deaths were reported this last season. 260. Highest number of pediatric deaths reported in any non-pandemic flu season since this became reportable. As we point out repeatedly, half those kids, majority of those kids, completely healthy, not even with obesity or whatever else you want to sort of throw to blame them for dying, and basically over 90% not vaccinated. And that's becoming a bigger problem. People are just not taking this seriously, not providing the kids with those wonderful life-saving protections. So. All right, RSV. We have a little bit of an update here. We have this article, Systemic Review and Expert Consensus on the Use of Long-Acting Monoclonal Antibodies for Prevention of Respiratory Syncytial Virus Disease. Armada Task Force, published in OFID. So Armada, what does that stand for? Armada, Advancing RSV Management and Disease Awareness. I like that, Armada. And so, as we've talked about repeatedly, the introduction of the RSV vaccines and the monoclonals really look like they've been a game changer. So the Armada Task Force, an international multidisciplinary expert panel, undertook a systematic review to develop long-acting monoclonal antibody consensus recommendations for RSV disease prevention in children. And the task force recommends for all infants aged less than 8 months, in the absence of maternal RSV vaccination, right? We talked about, where the moms actually get a vaccination during the beginning of that third trimester. So preterm infants also included, less than 37 weeks gestational age, aged less than 12 months, and children less 24 months with high-risk conditions. The long-acting monoclonal antibody administration is recommended. They say, although in RSV endemic countries, decisions should be made locally concerning administration year-round or with peak RSV incidences. So what is that last bit about? We're familiar here in the States that really it's about October. We start to see cases down in Florida. They kind of sweep up. But in the tropics, subtropical areas, I was kind of reviewing this to just sort of make sure this is what we're seeing. There can be year-round RSV. So it becomes a little trickier. They don't really get that seasonal winter peak that we get in some areas of the world. All right, COVID update. There's a storm on the horizon here, Vincent. If you look at emergency department visits in the United States, still very low, but we're starting to see an increase. And I will say that's something we're seeing here locally. I got a few folks in the hospital with COVID, some of them on oxygen, some of them having a bit of a tough time. And if you look at, I put the regional map, and everything's a little delayed, right? So it's a little bit behind the times. And as you'll see with this gray and our multicolored later on, some of the data we're always like 2 feet into it before we realize. But you could see up there in Alaska, down there in Alabama and Florida, really starting to get some pretty high wastewater viral activity levels.
Still, our curves are not, they're still flat. I'm still holding out for no summer peak.
Well, so this is going to be interesting because we're going to talk about an article, how to predict the future. So I have for...
Is it by Yogi Berra?
Yes, exactly. The hardest thing to predict is the future. And he didn't say half the things he said.
Oh, but it's fun to think he did.
But that's another one of his quotations, which is funny. I didn't say half the things that I said.
That's funny. Yeah.
Is that awesome? So, all right. So we have, following this out, all the way back to 2022, right? That's when we had our really last super high, very, very high peak. And then we've had this, up and down and up and down, and we had going back to 2023, we had a pretty nice long flat. Then it went up, right? It really didn't rise until end of July into August. Sort of zooming in on what's been going on here the last bit. Maybe some activity, but yeah, I'm going to give you that. I zoomed all the way in and, maybe, but again, you've got this gray period. So from 621 to 75, right? And we're here like getting, past the middle, getting into the end of July. So we're already like ahead of this. Maybe a suggestion that things are starting to go up. But here comes this article. Can we predict the future? The article estimated COVID-19 periodicity in correlation with SARS-CoV-2 spike protein S1 antigenic diversity, United States, published in Emerging Infectious Disease. So a couple comments here, and I enjoyed this article. It's a good article. Got some nice, fancy, multicolored figures. So start off by pointing out, as we've talked, a single winter peak annually is characteristic of several respiratory viruses, including seasonal flu, RSV, seasonal human coronaviruses. However, conversely, other respiratory viruses, including paraflu, rhinoviruses, enteroviruses, typically peak twice a year. So this is not unheard of, the idea of having the double peaks. I will say, as I mentioned, it's coronaviruses that fall in like flu and RSV. So we sort of have this expectation that eventually that might happen. Well, so here these authors report that their analysis revealed biannual COVID-19 peaks in late summer and winter. They say a pattern that is expected to persist as long as the rapid evolution of SARS-CoV-2 and cyclical S1 diversity continues. But then they note that a single dominant S1 sequence genotype was associated with both winter and late summer surges until November 2022. Since winter 2022-2023, a dominant genotype was also associated with a surge in virus activity during the winter season, but co-circulating variants with similar spike substitutions have been associated with late summer surges. So they've got this nice sort of pattern you can see, and the overlay really looks pretty similar to what we discuss each week. But I don't know, did you have any thoughts here? It's going to be interesting, right? They're predicting the future, and here we are, like entering the future.
Well, I think they're trying to associate genotypes with, these peaks. And that's what the peaks are human activity, in my opinion, right? That correlate with what people do at different times of the year. And I heard at ASV a very cool talk by a guy in Pasteur in Cambodia. And he says the peaks of avian influenza correspond with the activities that people do at different times of the year. And when you stop those activities, the disease peaks go away. So, the idea that you're going to find a magic mutation in the genome, I don't think so.
Yeah, I think that's been the idea. It's doing this because it's still like so rapidly evolving. But I don't know if it's still so rapidly evolving, actually.
Maybe, but it's too easy to blame disease patterns on a genotype, right? It's more likely the behavior of the host.
Yeah. And they sort of put in there, they're like, by the way, this isn't exactly true, right? They're already saying this dominant genotype, it works this way, but it doesn't work here. So,
I mean, we may see a little peak here because I think July 4th, there's a lot of activity. So we're lagging behind there. But I just do not think we're going to have the extent of peak that we saw last time. But we'll see.
Yeah. Yeah, no, it is interesting. We always talk about, it's like sometimes I watch the weather and then, if July 4th is like rainy and everyone's indoors, I'm like, oh my gosh, seems to be affected a lot by behavior. So we'll follow that. We'll see. We're gonna know in a couple weeks here. The bets are still in. Vincent thinks we're good until November? Is that what you're giving us? A flat till November? I think by the time we get to the end of August, it'll be clear that we're back up again. So all right, COVID early viral phase. So we have the article, Drug Treatments for Mild or Moderate COVID-19, Systematic Review and Network Meta-Analysis, published in the BMJ, all little letters, British Medical Journal. So really interesting, interactive, graphical abstract that I must recommend. I don't know, Vincent, maybe you should, everyone should go check this out because it's really cool. Like you move your mouse around and like different things highlight, like you move it to there's this circle and they've got all the different potential therapeutics, and then you click on it and you can you just hover over it. So it's really, I gotta say it's the best graphical abstract ever. So even if you don't care about the content, people, this is like just cool way of like sharing and communicating. So they start by stating that the current evidence supports 2 stages of COVID-19 disease. Oh my gosh, I feel like someone's been talking about that. An early phase marked by viral replication, that's when we get the non-severe disease, and a later inflammatory phase when we get severe disease. Have you heard of that model before?
There's this guy, Daniel Griffin, who said that like way at the beginning of the pandemic.
I think a bunch of us like published like in the spring, we're like, hey, this is what's going on and you should study treatments based on this paradigm. But all right, who knows? maybe I just got lucky. But the different pathogenesis and resulting severity of symptoms of each of these stages suggests different interventions are most effective at certain stages of the disease. Oh man, they're brilliance here. Consequently, our systematic review and network meta-analysis now addresses patients with mild or moderate COVID-19, also known as non-severe COVID-19, separate from those with severe 19, right? So this is that first week viral replication. In this publication, they're going to compare the effects of drug treatments for the first phase, separate publication looking at treatments for severe critical COVID-19. And this is going to be part of this series. So ultimately, in this, they looked at 259 trials, evaluating 166,230 patients. They compared standard drug, and they basically found two drugs probably reduced hospital admission. So nirmatrelvir ritonavir, right? So that's our Paxlovid, 25 fewer per 1,000, and remdesivir, 21 fewer per 1,000. Molnupiravir, systemic corticosteroids, may reduce hospital admission, but it's low certainty. Drugs for prophylaxis and antibody-based treatments, they're going to address separately. I want to point that out because I was like, oh, where's the data on the monoclonal antibodies, which were pretty impressive back when we were using them, but not seeing that covered here. So remember, one of the first things we recommend and sort of reinforced here, number one, Paxlovid, oral, way too expensive, by the way. But we have the article, extended nirmatrelvir ritonavir treatment durations for immunocompromised patients with COVID-19, EPIC-IC. So EPIC immunocompromised, a placebo-controlled randomized double-blind phase 2 trial published in the Lancet Infectious Diseases. So this is this thing that doesn't go away, right? People keep talking about like, well, maybe we're not giving it long enough. Maybe instead of 5 days, we should do 10 days or 15 days. Well, they studied this. This is a placebo-controlled, randomized, double-blind phase 2 trial that enrolled non-hospitalized immunocompromised individuals aged 12 years or older with symptomatic COVID-19 from 73 sites across 9 countries, right? So these are immunocompromised. Like if someone needs longer than 5 days, this seems like it would be the right group to look at. Participants were randomly assigned, so it's a 1-to-1-to-1 to get either 5 days, 10 days, or 15 days of Paxlovid. Primary endpoint was proportion of randomly assigned and dosed participants with sustained SARS-CoV-2 RNA concentrations below the lower limit of quantification from, this was interesting, from days 15 to 44. Kind of curious why they sort of start at day 15. We'll look at the actual data. Secondary endpoints include the incidence of viral rebound after the end of treatment up to day 44. Safety, a secondary endpoint, was assessed in all randomly assigned participants who received at least one dose of the nirmatrelvir ritonavir. So put in the bets. Everyone's ready. So is it 5? Is it 10? Is it 15? And they tell us no difference was observed between the 3 treatment durations in the primary endpoint. Extended nirmatrelvir ritonavir treatment beyond 5 days resulted in a nominal improvement in the frequency of the viral rebound, generally well tolerated. But let us take a little closer look here, right? So we've got this figure. The primary endpoint was reached in 61.5% of the folks in the 5 days, 70.8% in the people who got 10 days, 66% in the people that got 15 days. And then they've got these graphs where you can actually look at this viral rebound. And they're basically going to give us different numbers here. The big thing here is going to be wide confidence intervals. So they're going to say viral rebound occurred in 17% with the 5 days, 2% in the 10 days, 2% in the 15 days. So from gross numbers, right, you look at that, you say, seems a little bit different. And there were 2 COVID-19 related hospitalizations reported both in the 5-day treatment group.
It's probably not significant, though.
I think that was the problem. Yeah. So, these were not significant differences. Yeah.
It's very interesting.
It is pretty interesting. So I'm sort of give everyone the punchline, but also let people kind of look a little closer at the data here. You look at the 5-day group, and the severely immunocompromised. Those are really the only ones that we're seeing kind of maybe a little bit of higher nasopharyngeal viral RNA going out farther. You get to this 10-day, this 15-day group, they're really indistinguishable from the non-severely immunocompromised. So, I think we're still left with the 5 days, but it does sort of raise, some situations where, maybe severely immunocompromised, you might consider this. But again, it kind of becomes this issue about what is your end game, right? Are you trying to keep people out of the hospital? Are you trying to keep people, from not or getting through this? So, all right, number 2, remdesivir, right? And that was another one of the drugs that stood out in this meta-analysis. We still in the list in some parts of the world. We've got molnupiravir, we've got convalescent plasma. Not a lot of big changes during that second early inflammatory phase. We'll look forward to the big analysis of that, but still steroids, right time, right patient, right dose, anticoagulation, pulmonary support, remdesivir if we're still within the first 10 days, immune modulation. And that's going to bring us into the long COVID late phase, PASC. It's interesting. I was on NPR this week, and I was supposed to talk about long COVID and measles. Basically, I talked about long COVID and then just a little bit at the end about measles. But there was a nice special on NPR this last week, and you can go and listen to it as well. Just people sharing stories, people pointing out the fact that the current administration does not want to talk about long COVID. It's only about infection fatality rate. It is not about people that got sick and developed diabetes, had heart attacks, had strokes, have this debilitating, ME/CFS type presentation. But there are folks out there. We're still seeing them, unfortunately. Vaccination, tremendous way to reduce your risk of that. But what about therapies? So we hear from InVivid, that InVivid and leading researchers form SPEAR, Spike Protein Elimination and Recovery Study Group. I like that, SPEAR, to assess the effects of monoclonal antibody therapy for long COVID and COVID-19 post-vaccination syndrome. So just before I go into what they're going to say, just what's the concept here? So the concept here is the idea that there might be persistent antigenic stimulation of the immune system, right? So the whole idea that there's still spike protein, maybe a monoclonal antibody combined to that spike protein, clear that out, and you're going to reduce the ongoing immune activation. That's the idea. There's people, as we've talked about, who have this idea that, there's ongoing viral replication. We already did a multiple, day, multiple length, multiple studies looking at highly effective antivirals, right? Thinking about my HIV days there, but can we just give them lots of Paxlovid and they're going to get better? That doesn't seem to work, but maybe there's some way to mop up antigen with monoclonals, right? So the SPEAR study group was initiated in response to a growing number of case reports and reported case series from across the U.S. suggesting actually folks getting PEMGARDA, PEMGARDA, right, that monoclonal that you get every 3 months, actually reporting that they're doing better. They seem to be doing better. So people with long COVID are getting this for protection, pre-exposure prophylaxis. And a lot of them are saying, once I started this, I started to feel better. So PEMGARDA is not authorized for treatment of long COVID, and these emerging reports are, as they point out, anecdotal, uncontrolled, but collectively they support this plausible hypothesis that prolonged exposure to the quote-unquote pathogenic SARS-CoV-2 spike protein or other forms of persistent viral material may drive disease in some patients. So they're actually going to go ahead, enroll folks, give them the monoclonals, and see if it makes a difference.
I think that's a good study, but just remember that just because there's persisting antigen in long COVID doesn't mean it's causative. It could be a consequence or just an association, right? So unless we have animal models where we can determine what's going on, we just don't know. It's an observation that may not mean anything.
Yeah, I agree. And it is. This will be exciting. And that's what we got to do. We got to keep looking because we got people out there, millions of people out there suffering. And so great that we still have ongoing trials, and it's great that even if the federal government is shutting stuff down, that we've got companies, we've got private individuals supporting ongoing research. So I think I mentioned last week that the first grant that was cut was the long COVID study at Columbia. My colleague Lawrence Perper, and but we've had private people step in. We had the Cohen family step in with several $1 million of support to keep our clinic open, keep things going forward. So, thanks to all the people that are stepping in there. And here, as we've been saying for, 5+ years, no one is safe until everyone is safe. And I just thank everyone for all the contributions. That's what drives us forward. I want everyone to pause the recording right here, go to parasiteswithoutborders.com and click that donate button. The small amounts help, the big amounts help. We're getting to the end of our Foundation International Medical Relief of Children fundraiser, May, June, and July. Not quite there yet. We've got to get up to a $10,000 to double that for that maximum donation of $20,000. So thank you for your support.
It's time for your questions for Daniel. You can send yours to daniel@microbe.tv. Janet writes, sends a link to an article in CIDRAP saying adults don't need tetanus diphtheria vaccine boosters, according to a study. The question from Janet is, what about the pertussis vaccine, which was included in the booster, the DPT booster? I think you said it actually wanes before the 10 years is up.
Yeah, so this is great, Chad. Actually, I'm glad you leave this link in because I see this and I'm like, I wish I had that. Oh, there's the link right there. So thank you. But so this is an interesting study. What basically we have this shot. So the diphtheria, tetanus, acellular pertussis in there now. And wasn't that many years ago, I still remember when we actually went from just the diphtheria, tetanus every 10 year to the diphtheria, tetanus, acellular pertussis, 3-shot. Now, the interesting thing in this study, and I think we've been sort of observing this for a while, is even though we say, oh my gosh, it's been 10 years, you need that next tetanus booster. I don't know how true that is. I think the tetanus protection lasts well past those 10 years. There's plenty of adults out there not getting their tetanus shot every 10 years. And we're not, people are not dying left and right of tetanus. We do see tetanus in the United States, by the way, and we see it particularly in senior individuals. We might see it in nursing homes. We might see it what's been way past those 10 years. So 10 years is probably, more than we need for the tetanus. Probably also, as we're seeing from the study, the diphtheria protection you get lasts more than 10 years. But what about the acellular pertussis? And as we've mentioned before, the recommendations are driven by this diphtheria, tetanus every 10 year, but the pertussis does not last 10 years. So, what we've tried to do is a few things, right? So, tried to target high-risk populations. So, mom during the first week or so of that third trimester, right, to protect the kids. Try to get all the adults that are going to be around the newborn, get them to get updated. We don't wait 10 years. So try to sort of modify this. But yeah, this is an interesting challenge. Like, how often should people get a pertussis update as we're starting to see more pertussis cases and pertussis deaths?
Jessica writes, what's the ideal timing of vaccines in the third trimester of pregnancy? I'm currently pregnant with my first child due late October. And so Jessica lists 4 different vaccines. So let's take them one at a time. All right. First, Tdap. My OB typically gives Tdap at 28 weeks, which would be early August. They also recommend that all caretakers receive a Tdap booster. Doesn't matter when they get it, as long as it's 2 weeks before interacting with the baby.
Okay, yeah, this is great. Let's go through this one at a time, and this perfectly dovetails onto the last one. So the Tdap, 28 weeks is great. In general, what we're thinking about with the vaccines, and we'll have maybe even more data as we get into the other vaccines, is ideal is beginning of that third trimester or thereabouts. You want to give mom yourself, congratulations, by the way, enough time to really develop a good immune response. And as we've learned, it's not just antibodies. There actually is a transfer of cellular immunity through breastfeeding as well, right? So I like the Tdap being, here it looks like we're kind of end of second trimester, right before the beginning of the third trimester. But those are the thoughts. Now, as far as the sort of the ring immunity that we try to produce for all the caretakers, again, 2 to 3 weeks before they interact with the baby, enough time to get that immune protection.
Okay, RSV. My OB says the RSV vaccine can only be given between 32 and 36 weeks from October through February. At the beginning of October, I will just be beyond 36 weeks, so maybe barely not eligible. However, if I have the choice, am I better off getting RSV vaccine or waiting until the baby is born and giving them nirsevimab?
Okay. So this is a good one. So couple things here, right? Is sort of head to head. What's better? Mom getting the RSV vaccine, the baby getting nirsevimab, everything, right? And in most places, I think it's sort of a financial issue. They say, hey, you gotta choose. So if you're going to choose the RSV vaccine, ideal is right at that 32 weeks. If you're just beyond 36 weeks, again, there's going to potentially be a lack of time, right? So we're worried that it's not going to give you enough time. The nirsevimab, the monoclonal antibodies, which we just talked about a review, really are pretty tremendous. So that's a fine option in a situation like this, where you might say, you know what, it looks like the timing's not going to work here. Baby's born, gets nirsevimab.
COVID, I'd like to get Novavax in the fall, but I'm not sure I'll be allowed to under the new rules since I have no underlying conditions other than pregnancy. I got Novavax last year, much better than the week-long reaction I got from Pfizer or Moderna. If I'm allowed to get Novavax, what would be the timing? Should I wait until a new booster comes out?
Yeah. So, this is, I'm glad you bring this up. Like, it's just not fair, right? I mean, there are a number of individuals who say, I'd rather get the Novavax because I don't want to be down and out for 1, 2 days, 7 days in your case, right? I mean, you got enough going on. So it's really tough that some, individuals are taking away our access to vaccines. Even after they said under oath, they would do no such thing. The timing would be ideally, as I mentioned, the beginning of that third trimester. Like, it's going to give you protection, going to give the baby protection. And there'll be a new formulation this fall. So I think that makes sense. Yeah, let's see what happens with with this court case, and are people going to have access to the vaccines?
And flu, should I wait to get the flu shot until October, or should I get it earlier in case the baby is born earlier?
Yeah, so that's always a challenge, right? We've got this this timing issue because you get the vaccine, you get it during that third trimester. Not only is it going to protect you, but it's also going to protect the baby. There is a period of time from the time you get that shot till the baby is born, as you're developing that robust immune response that you'll share with the baby. But there's a challenge here too, right? If you protect yourself and you end up not getting the flu, then less likely the baby. If you end up getting it too soon and then you end up with the flu, you're going to have certain protection for the baby, but now you're going to have the flu. And we've talked about that 15% reduction in protection as time goes by. So I think it makes sense for you to get that flu shot. Probably October. It's probably a sort of a good timing for protecting everyone in scenarios like this.
If I'm eligible for all 4 vaccines, do any of them need to be spaced out for effectiveness?
No, I think this would be fine. Go ahead and get them all. Never miss an opportunity to vaccinate or get vaccinated.
Got to catch them all. I don't know if you get that reference, Daniel. It's like a Pokemon. Very
Pokemon,
good.
Is that what you're doing?
Yeah. So I love this email because Jessica recognizes the value of immunization. She's not asking us, do I need to get this or is it going to harm me? But when should I get it? I love it.
Yeah, it's great email. And thanks, because it allows us to walk through all these decisions.
Okay, Eric writes, during last week's episode, you discussed a lawsuit against RFK Jr. filed by 6 organizations based on new vaccine recommendations, right? As a longtime listener who had his professional and personal queries about infectious diseases answered on air by Dr. Griffin on 3 separate occasions, I was delighted for the opportunity to return the favor in some small way when he sought advice from any listeners who are attorneys like myself to explain the potential outcomes. The lawsuit filed in the Federal District Court for Massachusetts is civil in nature and seems to vacate, i.e. overrule, the actions by the Secretary, seeks to vacate, and several other senior health officials that removed the COVID-19 vaccine from the CDC immunization schedule for healthy children and pregnant women. If the plaintiffs prevail, the court would find that the actions by the named officials were arbitrary and capricious and therefore unlawful, thereby leaving the previous CDC vaccine schedule in place, which recommends boosters for healthy children and pregnant women. So there's the answer. And Eric gives some notes that we'll put in the show notes for your reading. But if they win the case, then we have to go back to the old schedule.
And then children and pregnant women will have access to vaccines, which we really should not be taking away that choice.
Although I can imagine appeals and appeals and appeals and this dragging out, right?
Yeah.
While people are in danger of becoming ill and perhaps dying, the courts will take their time. Tony writes, I feel the need to provide a correction to the recent discussion of the possibility that the adjuvant used for Shingrix and the Arexvy vaccines, that's the end of the sentence, both use the AS01 adjuvant, which does not contain alum aluminum. In fact, AS01 is a lipid mixture forming micelles lipid nanoparticles that have intrinsic innate immunity stimulating properties that enhance the effectiveness of the protein antigens in these vaccines. I appreciate that RFK Jr. made a big stink about aluminum in vaccines being a great hazard, despite the decades of evidence to the contrary. Although he should not be able to use that track, I have no doubt that he could up with another crackpot hypothesis that would similarly attack such lipid adjuvants using his Dunning-Kruger genius. In addition, there are 2 more of the many single antigen vaccines that have proven to be effective despite this other trope of his. I might also add there's an adjuvanted flu vaccine used in the U.S., at least for geezers like me, 77 years old. This has been the vaccine I've gotten the last 3 years, Tony.
Yeah.
And finally, David. In the Attack on Science section of TWiV 1230, you mentioned a citation by a dubious June 2424 ACIP presenter, Lynn Redwood, who referenced a thimerosal study by Dr. Berman. She implied Berman's study found, quote, long-term consequences in the brain from thimerosal. A simple online search reveals what a gross misinterpretation, as you pointed out, this was. Dr. Berman, in Toxicological Sciences, Volume 101, Issue 2, February 2008, is co-author of Low-Level Neonatal Thimerosal Exposure, Further Evaluation of Altered Neurotoxic Potential in SJL Mice. The last sentence of the abstract says it all, quote, the present results do not indicate pervasive developmental neurotoxicity following vaccine-level thimerosal injections in SJL mice and provide little, if any, support for the hypothesis that thimerosal exposure contributes to the etiology of neurodevelopmental disorders, and provides a link to that article. I'm deeply disturbed by the cruel arrogance and ignorance that has hijacked our government, institutions, and media. I'm grateful that you and Dr. R are doing what you can to fend off the attacks on medical science. I hope I live long enough to see truth regain its predominance in our national conscience. And here's a quote from Tad Williams. Every time we tell a lie, the thing we fear grows stronger. Let us be the ones growing stronger.
Oh, thank you, David. That's a great quotation, great email. So thank you to all the folks writing in.
So, Daniel, they took this article, which the title is non-committal, right? It's just long-term consequences. And they twist around the conclusion, assuming no one's ever going to look at the article.
I think that's the way it works for them. They're like, look at this article, and they put it up there, and then they say, oh, the authors say that, but when I looked at the article, it's like, So they make stuff up, and they use this veil, this illusion that they're actually reading the literature. No, but they start off with their conclusion, and then it's, even before AI, it was just like simple Google searches, and there's the title, that makes sense, just throw that in. I'm not going to actually read that.
Right. I like this cruel arrogance and ignorance. That's TWiV, Weekly Clinical Update with Dr. Daniel Griffin. Thank you, Daniel.
Oh, thank you. And everyone be safe.